তারপর বায়োলজিস্ট ফিজিক্স সবাইকে একসঙ্গে বুঝতে পারছিস Let me put it in the pocket here. That is better. Yeah, otherwise, like that. Yeah.
Okay, we can start. Again, we resume our session have two more talks. Okay, so it's great to have Manoj, who's from my PhD talk also on the stuff that we'll be doing on membrane biophysics.
Thanks, Debayan and Sandeep, for organizing this and inviting me in this workshop. So yeah, my research is on plasma membrane phase separation, which is a little different than what we are typically thinking about currently about LLPS using proteins and nucleic acids, which has a lot of interesting chemical properties, such as they might be involved in cation pi interactions and different functionalities, pi interactions. But lipids, on the other hand, are not necessarily have all these diversities. Lipids are, like as we all know, it's an amphiphilic molecule. It has a head group, and then it has a long fat. But what I going to show here today is that the phase separation of these lipids in the plasma membrane plays big role in the functions of cellular signaling, and in this time the signaling that I am interested in, immune receptor signaling, that happens and all these measurements are done in living cells. So the immune signaling that I am interested in is allergy signaling that happens in our body. So in allergy signaling, what happens once we are exposed to an allergen, say for example brinjal or peanut butter or whatever, and then this particular receptor called Fc epsilon R1, this allergen binds to them, it gets clustered, and then a lot of processes downstream to this starts working. In the end, it releases some materials called histamine, and we get those small little blisters, red blisters, which are moieties packed of histamines, and then to inform you that you have been attacked by an allergen, and then you take an antihistamine medicine to reduce it, and you are good to go. So just focusing on the very early stage of this, what happens is that Fc epsilon R1 receptor binds to the antigen, it makes a complex, then this complex is not active. Active means that once this complex is phosphorylated, remember the antigen is bound to the outside of your cell and the phosphorylations happen at the inside of your cell. So, which means that antigen cannot directly phosphorylate the receptor. So, something must have happened inside to phosphorylate the receptor so that all these downstream processes can start. And this happens by in all biological processes. Phosphorylation is catalyzed by a molecule called kinase, and of course in biology everything must be reversed, because you take a medicine, it must be reversed, right? It has to be reversible, and there is always an antidote to kinase, which is a phosphatase. So they are all present there. Now the question is that we are putting here is that how this receptor, kinase, phosphatase, all of they are present before and after antigen, but this activity is happening only after antigen is present, right? Because everything is happening in the intercellular part, and they are all present in your body at all times. But the moment something is bound outside, it is doing something, and we start seeing this activity, or in other words, this balance is shifted more towards right, so that the signaling happens. And this is where the an attractive model that comes in is an LLPS model. So, and it is the LLPS of the lipid. So, what this model posits? It states that when you have unbound states, we have nothing, and then when the receptor is cross-linked with the antigen, it forms this cluster, and this cluster somehow assembles an ordered state around it. Now, when you said ordered state, I will be discussing a little more in the next slide. In ordered state, what happens is that there are lipids which has unsaturation in their acyl chain, in their tail, so they do not pack well, and then there are lipids which are saturated lipids, and they do pack well. So you see that if these lipids are put together, there is no packing defect, and as these two lipids they self assemble, there are packing defects. So this makes them more disordered, and this makes them ordered. Now you mix them, mix a lipid which has unsaturated tail, a lipid saturated tail, along with cholesterol which is ubiquitous in plasma membranes, and then you start seeing a unique behavior where cholesterol does break this long range attractive interactions in these saturated lipids, and also it induces some sort of order in this unsaturated species. So, giving rise to a liquid phase separation at the plasma membrane. So, this is what you see in the model system. So, once we get that, now the kinase, which is lin over here, this is a protein that is anchored to the plasma membrane in inner leaflet by a saturated tail. So, of course, the kinase likes to be present. So, it partitions into those liquid ordered region. So, now it has a more chance to see the receptor and the receptor tyrosine, and it can do its action. Okay, so, that is the model. So, now moving to the membrane lipids, membrane organization, you see that there are inner and the outer leaflets. So, this is the outer leaflet and the inner leaflet. There is a very diverse lipid composition, and if you, as I was talking about the tail unsaturation, you see that outer leaflet has a lot of saturated species, while the inner leaflet. So, when this x axis is lipid unsaturation, meaning that most of the lipids in the outer leaflet are indeed saturated. There is zero unsaturation compared to the inner leaflet, where most of the lipids are unsaturated. So, so that is one thing. The other thing is that is also experimentally tested, and it was found that outer leaflet, since it has a lot of less unsaturation or almost mostly saturated, it has a slower diffusion. As I said, it is more ordered, hence it has slower diffusion compared to the inner leaflet, which has a faster diffusion. Similarly, you can look at the other. So, this is the direct measurement of lipid order, and you can confirm that. So, what happens, as I was mentioning, that there are unsaturations over here, saturation, and then when you put them together, you can really create this LOLD like membrane. This has been tested experimentally by standard phase diagram. It's been long tested, DOPC, DPPC, and cholesterol, and all these are not in live cells. These are like synthetic lipid membranes. So, you can clearly see what we call nowadays a familiar word, droplet, right. So, you see over here, this is a fluorescence image, and of course, outside this coexisting region, you have a single phase, and as you go from a little bit inside this boundary or above the CSAT of all these three components, you start seeing phases. So, go from 4, 5 to 7, you are increasing the cholesterol content, 4, 5 to 7, 4, 5, 7, you are seeing more and more phase separation, 4, 6, 8, you are getting more saturated lipid content, 4, 6, 8, you are also getting more phase separation. All right. So, this is all good, and again on them people measure diffusion directly and they say differences. So, yes, both of them, the tracer molecule, they do diffuse in both phases, hence they are both liquid, but they diffuse at a very different rates. So, one is in the order of 0.1 micrometer squared per second, one is about 3, 4 micrometers squared, so order of magnitude difference, and which means that both of them are liquid, but have a very different diffusivity of the tracer inside those domains. So, these are all good in synthetic membrane. The problem starts when you start looking into it in live cells. So, these are live cell images. So, this is your cell, and in the cell you do not see any phase separation. But what are those? So, we create chemical stress into the cells, and the cells set off some vesicles directly from the plasma membrane. Now, the moment the cells are shedding this plasma membrane, the vesicles from the plasma membrane, you see that there are phase separation. So, in other words, the hypothesis is that maybe the plasma membrane do phase separate, but they are not resolved by our optical microscopy. But when you are trying to get the vesicles directly from the plasma membrane, or in other words, the cell is totally in out of equilibrium. There are so many things happening in cells. They are full of ATPs, there are actin cytoskeletons, there are other things that are controlling the behavior of the plasma membrane. But now you are isolating a vesicle directly from the plasma membrane, which does not have all these complexities, and you see now you clearly see the phase. So, in other words, it is allowing this small little droplet to coalesce so that they become optically resolvable. So, all in all, this image states that plasma membrane is likely have the capacity to phase separate. And indeed, when we took these vesicles directly out, and here what we are doing in the cytoplasmic leaflet, we take different probes, saturated lipid probes, unsaturated lipid probes, and you see that they clearly phase separate. Okay? So, just now, which means that the inner leaflet of the plasma membrane, when this is isolated from a living system, they do phase separate. Again, the diffusion behavior shows that they are liquid, but they are in a very different state. So, now the question, so this gives us hope that we can test whether this phase separation impacts our interactions between the kinase and the receptor, as I was hypothesizing in the couple of slides back. So, what do you see now? First, we start testing by standard imaging whether or not we see the nanoclusters. Yes, we do. So, you just stimulate the cells. Before that, there is almost featureless membrane. This is the receptor that we are imaging, and after you add the antigen, you see the small little dots, which means the receptors got clustered. Then, if the signaling happens, meaning that the action between lean and kinase happens well, then the Syk kinase, which was at the cytosol, that will go and bound to this receptor, and you will start seeing them at the plasma membrane in a similar punctate features, which is what you see, which means that the signal has been transmitted from outside to inside, and Syk got to know this, and it is recruited to the plasma membrane, which also means that there must be an intermediate step in which the receptor got activated by the kinase. So, if there is a clear interaction between the kinase and the receptor, we are supposed to see similar punctated structure of lean kinase as well. And why is that so? Because if they are complexing, if they are talking to each other, if they are forming colocalizing at all times, then I am supposed to see the punctate structure, just like I see for the Syk, but I don't. It means that, but given that I am already seeing Syk is recruited to the plasma membrane, it means that this interaction had happened already. But this is a featureless image. It means that this interaction must be very transient. And this is also observed when we do the standard FRAP experiment, in which what you do, you just bleach a spot into the plasma membrane and you see how the molecules are recovering. So if a molecule or a protein is immobile, it doesn't recover because it doesn't diffuse anymore. So that's the case for the receptor, because it was bound, it is clustered, and it is mostly immobilized. Lean, it's not the case. It was mobile before receptor clustering and after receptor clustering, meaning that it interacts with the receptor, but it is not strong enough to make it immobile. And this is how we see it. So let's say this is the lean molecules, the black ones, and it's the lean trajectory before and after clustering. So before clustering, there was nothing; it is not interacting with the receptor. But now the receptors clusters are formed, the larger ones, and the lean molecule are going there, interacting with the cluster for a while, and then coming out. Right? There are some lean molecules which will not interact with the cluster either. So now if someone makes a measurement in these particular regions, then they probably would see a little bit decrease in the diffusion of lean, because it is interacting for a short while. But if someone is making a measurement on the other places like these ones, you don't see any changes of lean diffusion, because they are not interacting. So in FRAP, I don't get to see it, where I should bleach my spot. I don't know. So I just bleach anywhere randomly, and then I collect the statistics and I see, oh, nothing is happening, simply because we are not collecting enough. Maybe if we collect tens of thousands of data points, statistically you will probably be able to see that. And this is exactly what we need to do. And we came up with this method that we do. It's called FCS, fluorescence correlation spectroscopy. This is another equivalent measurement of FRAP, but now you can get correlation function or diffusion measurement at each spot on the plasma membrane. These spots are very small, like 200 nanometer spot, and your membrane is 50 micron by 50 micron. So in one go, you get 10,000 data points. Once you do that, what happens? That indeed you see that this is the cumulative distribution of diffusion constant of lean, and the red one is before antigen. After stimulation, you see a clear red shift, blue shift in its diffusion constant. It means that the diffusion indeed decreased. Now this decrease is only about 10%, which is what you expect, because remember I was telling the interactions is very transient. So if it is a transient interactions and you are making a bulk measurement, you are not getting to expecting a huge difference, right? And this is and the good thing about doing this enormous measurement, so we are talking about 50,000 data points. When you do this scale of measurements, you start picking up small differences. Of course, you now have the other problem that you end up getting false positives. So you do run a lot of statistical tests, which I'm not going to talk, but a lot of different statistical tests to and complementary experiments to test whether this is real or not. So that is different. That is, all these controls had been done to justify that this is a real 10% difference in the diffusion constant. Now this is good. We got to see for the lean. Now the next question is, my original hypothesis was somehow after clustering there is a stabilization of ordered and disordered region. Around the cluster there is ordered region. So how do we test that? So we again go back to our standard probes, which is a saturated probe and unsaturated probe, and let's say this is their diffusion constant to start with. Of course, the saturated probe will have a little bit slower diffusion than the unsaturated probe. Now if the stimulation leads to a stabilization of the ordered disorder, what will happen? This probe, the saturated probe, will spend more time into this ordered domain, because the ordered domains are now stable, slightly more stable. So it spends more time in the ordered region, so diffusion further decreases. Now that is what I was telling, or what the first speaker of today described as above CSAT. So there is dense region in one place, in exchange of a dilute region elsewhere. So, if my ordered lipids, the saturated lipids comes closer to the receptor, that becomes ordered and saturated rich region. Elsewhere, it becomes unsaturated rich region or saturated diluted region. So, there where the unsaturated lipid prefers to partition, it goes there and spends more time there, its diffusion increases. So, if my measurement shows that PM goes, which is the saturated lipid probe, PM goes down, its diffusion, GG goes up, I know that there is an the LLPS has been stabilized. This is exactly what we observed. Again, these are the two probes I am showing now. I have a series of probes, saturated, unsaturated, where we saw the same thing. Next, what we did, okay, so we proved that we developed a method that detects very subtle differences. That method also detects that there is a stabilization of order and disordered phase separation in the plasma membrane. Now, the next question, does Lin kinase utilizes it? Because remember, Lin kinase has all the protein motifs that directly interacts with the receptor, that can, right. So, what we did for that, we created a mutant in which you see this lipid part, lipid component of the whole protein is mutated, and now we added another component that is disorder preferring. So, while all the other motifs remaining the same, meaning that this Lin kinase remains functional, okay, only thing we changed, it is membrane attachment. We make it order preferring Lin to a disorder preferring Lin, and there are tests to show that this become disorder preferring. It is just you add mild detergent to the system. If it is entirely soluble, we call it disorder preferring. If it is insoluble, we call it order preferring. Now, what you see here, we lost this decrease, stimulator decrease of Lin. In fact, it exactly resembles what a disorder preferring probe would do. It slightly increases, not as much as the other probe I showed, but it slightly increases the diffusion. So, it is spending more time in the disordered region, even though it has all the protein interacting motif. So, this is the example to show that our LLPS is implicating biological interactions. Moving even more detailed functional interactions, what we did here, we reconstituted in a system the kinase, phosphatase and the receptor, and we look at plus or minus Ag and see whether the receptor is phosphorylating or not. So, when wild type kinase, wild type phosphatase and the receptor all are present, we see clear phosphorylation after antigen. When we take S15, which is the Lin mutant, we do not see anything. We do not see the change in phosphorylation, and of course, without Lin, no phosphorylation. This is the control, negative control. Without Lin, just with the phosphatase, no phosphorylation happen. So, this is to show that just by changing the lipid anchor, not the protein interacting motif, we can actually change its function, and that is why I just mentioned that liquid-liquid phase separation is impactful in the functional of membrane. Okay. So, this brings me to this another state of this another part of this project where we are looking at so initially in the resting states everything was present, and in the stimulated state LOLD is stabilized. So, if it so happens, if I change anything in the resting state, I increase or decrease the order of the resting state, does it impact on the stimulated changes? Now, I would say yes, because in the end of the day, it is how much stabilization you are making over here that dictates how much time Lin will spend over here. If the ordered region are not so stable, Lin will not spend so long in there, and it will have less chance to talk to the receptor, and the vice versa. So, I would say that if I make my system more ordered to start with before antigen stimulation, I will see a difference, or less ordered, I will see an opposite trend. So, so this is what we did in this set of experiment. What we did here, we come up with an idea of exchanging the outer leaflet by a leaflet by some lipids of our choice, and this lipid can be saturated lipid like DPPC or DSPC, or can be an unsaturated lipid. So, once we do that exclusively on the outer leaflet, we see changes happening in the inner leaflet, and this is how the changes are made. You see that when again the probes are same, PM and GG. When we add the POPC, a unsaturated lipid, we see a diffusion to increase, and when you have the saturated lipid, the diffusion decreases. It means that if you add unsaturated lipid or disorder promoting lipid, you see disorder in the inner leaflet. Remember, all my reactions are happening in the inner leaflet. So, I cannot perturb the inner leaflet lipid. So, that I have to keep intact. All I am doing something in the outer leaflet so that I can induce order or disorder in the inner leaflet and test whether it matters. So, this is what we do for PM, and similarly when we look at GG, we do the same thing. So, there is a global change, unlike the last one when we see the somewhere it is ordering, somewhere it is more disordering. This time, since I am changing the outer leaflet in a global scale, I am seeing a global impact. Either the membrane is entirely becoming ordered, or the whole inner leaflet is becoming disordered. And once we so my system is now prepared before stimulation in a resting condition. The system is prepared either disordered or ordered. And now I add my antigen and see what happens to this sick recruitment. So, sick recruitment is our functional test. So, higher or more sick recruitment or faster sick recruitment testifies that it is over signaling, or if it is less sick recruitment or slower sick recruitment, we call it hyposignaling. Okay? So, now we see in here that you see that, so initially we have the control that is in red. If you have POPC, which is disorder preferring probe lipid, you see that there is a decrease in the stimulated sick recruitment, meaning that again how my system is prepared, how my resting state is prepared, that dictates whether my stimulation will be optimal stimulation or hyper stimulation. So, with this we went to this talk and we showed that the plasma membrane lipid phase separation plays a big role in the biological context, and in principle there is a functional impact of this LLPS, and again it signifies the importance of the resting state. If your resting state is poised to respond, then you will get optimal signal. If it is not, if it is somehow perturbed, then you also get dysfunctions. Okay? With that, I would like to thank everyone who contributed, especially my collaborators and my lab members, and thank you for your attention.
I had two questions.
Yeah.
The first is, how do you deal with enzymes that flip lipids across the two leaflets?
Yeah. So, the lipid flipping that we like, the time scale of these measurements are very short. So, we do antigen stimulation, and within 10 to 15 minutes this is all done. Lipid flipping, like the real, like the PS, phosphatidylserine or PC lipids, that flipping happens in the hours time scale. So, we have tested that whether during this process of our measurement time scale, whether the lipid flipping is happening or not, and we observed that lipid flippings are not happening, and those enzymes, they remain active. So, they are doing their job, but it is not
on a slow time scale.
Yeah.
The second question, which is, I wonder if you have heard of T cell independent B cell activation of naive B cells, where the major histocompatibility complex is not involved.
Yeah.
For instance, non-proteinaceous antigens like lipopolysaccharide.
Yes, yes.
Where the epitope is actually repeatedly displayed
Yeah.
on a very long chain antigen,
Yes.
and that ends up cross-linking the
Yes.
immunoglobulins
Yeah.
receptors on the cell surface.
Yeah.
And I, from when I first heard of this, of course, liquid-liquid phase separation was not being talked about, but just listening to your talk made me wonder whether T cell independent B cell activation
Yes.
actually involves a clustering of receptors and any kind of LLPS.
Yes, the answer is yes, because and I know a couple of people who are working on that, exactly on that. The reason why I am saying this yes is because all these components like lean, seq and other things and the receptor and the multivalency, these are pretty much similar between B cell receptors and the mast cell receptors, the epsilon receptor. The differences is on how you present the antigen. Now, in the T cell independent B cell signaling, the presentation of antigen is very similar to that of mast cell. So, that is why I think, and when I talk to those people who are working on that, they also see similar behavior, the LLPS of the lipids playing a role in there.
Yeah, very nice talk. So I had 3 questions. So one being, so how fast are the dynamics of the formation of these? You said within 10 minutes you see.
Yes. So that is exactly what we are now testing. It happens in the time scale. So after you stimulate, it happens in the time scale of 5 to 8 minutes. So that's what
And if you keep it for longer, do they coalesce to become like bigger?
That is what we don't see.
Okay.
And this is why this goes back to that small image I showed, that there are ways to stop from over coalescence, and these ways, one of the major ways is actin cytoskeleton, because these are meshwork and it doesn't allow you to grow. So if you deplete actin cytoskeleton, it is not optically resolvable, but if you do super resolution microscopy, and I have some data on that, where you could see that they are phase separating in when the activity of actin cytoskeleton is diminished.
And if you increase the orderness in the leaflets, do the size of these things increase? Have you looked at that?
I would think yes, but I never
It's
tested
still
that.
diffraction limited.
It is still diffraction limited. I don't see it definitely. And one thing you have to also note that just by changing this order disorder, you are not changing the order on the resting cell, you are not stimulating anything. So you see no recruitment. So they are not necessarily happening at a larger length scale that the seq is fooled to be recruited. So that doesn't happen.
But even at smaller length scale,
I do believe that it is there they are becoming larger. I do believe, but I haven't resolved. I don't know how to resolve either. Yeah. Yeah. Yeah.
Not very different.
It is not very different, but what we did is there are many various ways we tested. So what we did, we take these measurements and we just then take subset of 10% of the data, right, in both conditions, only 10% of the data, and then we do all sorts of rank correlation analysis to say whether or not they are difference, only with 10% of it, and we do repeat it like 100,000 times and see how many times it is giving us totally different. And we say they are different if and only if after doing 1,000 times, every time it states the p-value is different using rank correlation.
Well, I mean, generally I have suspicion about most statistical methods.
Yeah. So
Because there are issues with
there are issues in,
assumptions.
there are fundamental assumptions to
Like
it.
when you say p-value, you are assuming that the distribution
Is Gaussian? No, I am not doing so. That's why I said I did rank correlation, and it is.
But you believe that it is different. I mean,
And because I have experimental tests, plus all this, as I said, it's only the 10% of all data I took from both population and see whether they are different or not. And then this is done 1,000 times, and every time if I see that there is always difference, it is giving, that's the benchmark. But again, like some days later, there will be some better statistical test will come, and I will run through again through my those tests with my data.
Okay, another question is the distributed position that
Yeah.
do you actually see like linear, like when you plot the square average in the time, do you see it to be linear or
To the range I am talking about, the length scale, yes, it is linear. Yeah.
like
The length scale, remember, I am not looking at single molecules. I am looking at a length scale which is about 200 to 50 nanometer big. So at that length scale, the anomalous diffusion oftentimes are not observed. So I am taking all the possible anomalies together, and they are averaged out at that length scale. Yeah. Thank you.
the next speaker is Shivananda Das.
Is it green? Am I audible at the back? Yeah. Okay. So first of all, I would like to thank Devayan and Sandeep for inviting me to this interesting meeting. So what I am going to talk about today is self-organization of charged macromolecules, particularly of intrinsically disordered proteins and polyisotions. So part of the talk might not be directly about biomolecular systems, but still it is relevant for biomolecular condensation. So to a physicist like me, the biological cell is like a Coulomb soup, because all the typical cellular compartments or organelles constitutes of various types of charged molecules and also electrolyte ions, and that's why the fundamental forces that are operative in this dense crowded cellular environment arise from the interaction between different charged components, and also from the weak range van der Waals type interaction, through which the different components communicate or interact with each other. And how does this communication and cooperation happens, and what are the factors that that modulates this communication, that is of fundamental interest to us. Now, the speakers before me have already, like, in great detail introduced what biocondensates are, so I won't go into much detailed introduction about them. So we know that biocondensates are basically hierarchical structures of different types of proteins and nucleic acids, and in our study we were particularly interested in the biocondensates of intrinsically disordered proteins, which are known to self-assemble into membraneless organelles or droplets through liquid-liquid phase separation. Now, but overall the phase behavior of this IDPs can be quite complex because of the confluence of many different attributes. For example, there are entropic contribution due to the chain connectivity of these macromolecules, and then we have electrostatic monopole and dipolar interaction, and also hydrophobic interactions between the different segments. And even the overall sequence of these macromolecules are expected to have an effect on their overall phase behavior. So to explore along this idea, we considered the particular IDP alpha synuclein, and then investigated its aggregation mechanism. So this here you can see is the sequence of this protein. So if you look closely, so this is the N-terminal segment, which has a net slightly positive net positive charge, but if you look closely, you will see that there are many pairs of positive and negatively charged residues in this segment, and that's why this segment has a overall dipolar property. And then there is this intermediate neck region where you will typically see the different secondary beta structures. And finally, there is this C-terminal which has a very high net negative charge. And because of the concentrated charges at the termini and the overall hydrophobic center, this protein typically shows the characteristics of an IDP. Now, this is actually a well characterized protein in experiments, and what we know from experiments is that in bare solutions of the protein, droplets are very hard to form, but when you have Crowder molecules like PEG, you will see that droplets or membraneless or biocondensers very readily form in the solution of this protein, and these droplets over time can transform into more viscoelastic and solid like amyloid fibrils, which are believed to be connected to the neurodegenerative disorder Parkinson's disease. Now, in our study, we did a detailed detailed investigation of the structural organization in the solution of this protein using coarse grain simulation, and that's what I'm going to talk about today first. And then later I will show some extension of this study to how does complexation with RNA affect this structural organization. And finally, I will also talk about one important aspect of not just this IDPs, but other charged macromolecular systems, which is the effect of sequence. So, so as I said that we basically do a coarse grain simulation, and the coarse graining is at the level of one residue, at the level of the residue, and obviously we have the aspartic acid and glutamic acid which are negatively charged, and lysine, arginine and histidine are positively charged. So we consider electrostatic interactions between these residues. And in addition to that, we have basically total 5 different force fields to account for the many different types of non-bonded and bonded interactions between the different residues. And so in the beginning, we characterized the different structural properties in comparison with experiments, and here I would like to particularly show the results for the radius of gyration, which we find to be quite good agreement with experiments, and even we observe a similar trend as in experiments where the radius of gyration increases with increasing ionic strength. So what happens here is that what happens is that at lower ionic strengths, such kind of self-complexed configuration with contacts between the dipolar N-terminal and the negatively charged C-terminal are quite prominent, but as the ionic strength increases and the electrostatic interaction gets screened, such kind of configuration becomes less probable, and instead the protein acquires a more extended configuration which leads into an increased radius of gyration. So from this observation at the single chain level itself, we could anticipate the important role electrostatic interactions may play in the bulk solution. So let's see what exactly happens in the bulk solution. So these are simulation snapshots for bare alpha synuclein solution. So as we increase the concentration of the protein, so initially at very low concentration we observe many dispersed protein chains, and we refer to this phase as the fluid phase, but as the concentration increases, aggregates of intermediate sizes emerge in the solution, and finally we observe this large percolated network in our system. And so we basically characterize the size of the different aggregates or self-assembled structures by calculating the cluster size distribution. So particularly for this largest concentration of 10 millimolar, you can see here this distinct peak towards right, which corresponds to the large percolated network that we observe in the system, and it from this plot we can see that it almost contains almost 90 to 95% of all the protein chains in the system. So now that we had some idea about like what kind of assemblies we appear in the solution, So the next question was how are the different protein chains organized, or what is the correlation between the different protein chains. And to answer this, we looked into many different quantities. So one of the quantity we looked at is calculated is the static structure factor, which is accessible in experiments through different scattering measurements. So typically in case of a simple liquid-liquid phase separation or macrophase separation, you would expect the peak for the structure factor to appear at the thermodynamic limit of Q0. But now for the protein chains in the percolated network in our system, we actually find that there is a characteristic peak at a low Q value, which is the characteristic, which is the signature of microphase separation. So actually there is a preference for a characteristic length scale. So to explain it in other words, so basically you have the protein chains organized into small microdomains which are interconnected. So this percolated network is formed of many interconnected microdomains. Then we also looked into the interchain contact map to get an idea about like which residues are interacting with has more interaction with each other, and which is driving the organization into this percolated microstructure. So you can see here that there are a large number of contacts between the N-terminal and the C-terminal residues. So as we saw in the single chain case, that how electrostatic interactions are quite important determinant of the structure. So here also we actually see that the electrostatic interaction between the dipolar N-terminal and the negatively charged C-terminal is what is driving the organization into this percolated microstructure. Now since electrostatic interactions are important, we also did a very detailed characterization of the effect of the electrostatic interactions by varying ionic strengths. So here I want to particularly show you the results for the static structure factor. So here, so what we find is that at lower ionic strengths, when the electrostatic interactions are much more stronger, the peak is slightly shifted towards right. So in other words, the characteristic length scale that appears in the system is smaller at lower ionic strengths, or in other words, these microdomains are much more compactly packed inside at lower ionic strengths. And since alpha-synuclein is very well known as a RNA binding protein, so the next obvious choice for us to characterize this electrostatic interaction was to check how does complexation with RNA affect the structures that we observed in earlier in the Bayer solution. So for this study again we do consider a coarse grain model for the RNA, where each nucleotide is described by 3 beats, and we make sure that the coarse graining length scale is consistent with our the coarse grain model for the protein. And again here we have many different interactions to consider, many different force fields to consider the interaction between the different nucleotides. And particularly for the RNA protein interaction, we obviously have the electrostatic interaction between the negatively charged phosphate groups and the charged residues of the protein. And in addition to that, we consider some interaction between the guanine and cytosine nucleotides and the residues of the IDP, because from experiments it is known that alpha-synuclein has a propensity, binding propensity for these 2 nucleotides. That's why we consider these additional interactions. Now with the inclusion of RNA, again at first we looked into how does the structural property of a single chain looks. So here again you can see the radiusal gyration as a function of ionic strength. In absence of RNA, earlier I showed like how with increasing ionic strength the radiusal gyration increase, but now when the protein is complexed with the RNA, we see actually an opposite trend. So now the radiusal gyration decreases, increasing with ionic strength. And so for now if you look into the contact maps, so even though I am showing here the contact map for this particular ionic strength, but for all the ionic strength, you will see that there are almost no contacts between the N terminal and C terminal. So what happens is, so if you look into this snapshot, what happens is that the complexation with the RNA happens through the dipolar N terminal, and the negatively charged C terminal doesn't participate because of strong electrostatic repulsion. Only when the ionic strength is high and this repulsion is screened, this C terminal is able to be more compact and come close to the rest of the body, which leads to a reduction in the radius of gyration. And not just for a single chain, but when we introduced RNA in the bulk solution of the protein, there also we observe a structural reorganization. So in absence of RNA, we earlier saw that how there are large number of contacts between the N terminal and the C terminal. But now when we introduce RNA and increase the percentage of RNA in the solution, these contacts disappear, and instead we see a large amount of contacts between the N terminals in the N terminal residues of the protein. So basically what we find is that RNA is favoring formation of cross links or contacts between the N terminal residues of the proteins in the bulk solution. And then in this case again to get an idea of the different aggregates or the size of the aggregates that appear in the solution, we calculated the cluster size distribution. So at higher concentrations, we observe the usual percolation peaks, and these percolation peaks actually appear at even earlier concentration with the in presence of RNA. But now at lower concentration, an even more interesting observation appears. So in absence of RNA, this distribution curves were monotonically decreasing. But now when RNA is present in the solution, this distribution curves becomes non-monotonic with a peak at an intermediate cluster size value. So RNA is facilitating formation of small microdomains, in this case even at much lower concentration. So that was basically an interesting observation when we included the RNA. And sorry, so basically to give you a summary of the main conclusions from this part about this alpha-synuclein IDB solution. So first of all, we saw that at higher concentration a percolated assembly appears which has a microstructural organization, and then with the inclusion of RNA we find that actually RNA reinforces the microphase separation even at a much lower concentration. Now let me go to the last section of this talk, which is the sequence effect, and so this is quite important for many charged macromolecular systems. So initially I mentioned that for IDPs you will quite often see pairs of positive and negatively charged residues close to each other, or even for other charged macromolecules like polyisotianes, for which you will see that each of the repeating unit is explicitly dipolar. Now to understand the effect of the dipolar interaction, or specifically the effect of dipolar sequence, we considered a coarse-grained model of polydipoles where each of the monomers were described by 3 beads, out of which the backbone beads were hydrophobic, and the side chain beads could be hydrophilic and could be dipolar. So before going into the effect of sequence, let me share some results about the homopolymer, where basically you have all the all the monomers to be dipolar. So in this case, when we varied the temperature for different combination of the dipole interaction and hydrophobic interaction strength, so by varying the temperature we could observe the coil to globule transition, and from those transition, when we extract the theta temperature, what we find is that the theta temperature shows a linear dependence on the hydrophobic interaction strength and the dipole interaction strength, and such a p-square dependence on the dipole moment tells us that there can be 2 possible combinations of dipolar alignment, basically the dipoles which are in close contact. But here, so this is a snapshot of the globule that forms at lower temperature. Here you can clearly see that actually antiparallel orientation is the alignment is the main dominant mode of organization for the dipoles, and not just in this single chain level. Actually, when we looked into the gelation of these polydipoles, so this is a typical phase diagram of an associative polymers. So to study the gelation, we constrained ourselves at temperatures above the critical temperature, and so this is a phase diagram of this polydipole system, where you can see this percolation line separating the solution and the gel phase. Now even inside the gel phase, when we did a characterization of how the dipoles are oriented, so particularly we looked into the angles the dipoles form. So there we see that again there is a preference for the such antiparallel alignment instead of such a parallel alignment with stacking on each other, even though this might have reduced the energy by a factor of 2, but is not but is hindered because of steric interactions. And such a p-square and this p-square dependence observation is particularly important, because typically when you consider mean field theory for this kind of polydipole systems, a randomly oriented dipole is considered, which leads to a p to the power 4 dependence. But what we showed from our simulation is that such a approximation is not always true. So this was for a homopolymer case, but let me actually move to the part which I promised that I will talk about, so which is the sequence effect. And since we are now interested in the effect of sequence, so what we did is that we considered, instead of considering all the bits to be dipolar, we considered only half of them to be dipole and the other half unpolarized, and then we distributed the dipolar repeating units in blocks of the different sizes along the backbone of the polymer. So for example, in the sequence S1, every alternate bit is a dipole, and that's why for our polymer chain with degree of polymerization 256, we have total 256 number of blocks, whereas this sequence S8, in this sequence you have one half dipolar and other half unpolarized. Now for these different sequences, again when we varied the temperature, we could observe the typical coil to globule transition, but interestingly in this case the theta temperature for this transition showed a sequence dependence, where this theta temperature monotonically decreased with increasing number of blocks, or in other words, for more homogeneous distribution of the dipoles, the theta temperature was lower. And actually this is since the theta temperature is correlated with the critical temperature of the system, so this indirectly tells us that actually so for the more homogeneous sequences, you will observe the liquid-liquid phase separation even at a much we at a much lower temperature to observe the liquid-liquid phase separation. And so we have this such a dependence, but why is it so? To get a feeling about that, we explicitly calculated the free energy difference between the globule and the coil state for each of the sequences, and what we found is that for more blocky sequences, the free energy difference between the globule state and the coil state is large, and that's why to make this jump from the globule to the coil state, you have to be at a higher temperature, and that's why the theta temperature is higher. And actually this sequence dependence in free energy is inducted by the dipole-dipole interaction energy, and which you can see here. So these are the dipole interaction energy as a function of temperature for the different sequences, and you can clearly see that for S1, which is a more homogeneous sequence, in the globule state, which is at the low temperatures, this dipole-dipole interaction energy is much lower compared to more blocky sequences like S5, and so that's how like this sequence dependence is inducted for the different sequences. And with that, I would also like to basically summarize the two main conclusions from this section, which are, so first of all, for homopolymers, we found that that antiparallel orientation is basically the main determining factor for the structural organization into a gel, as well as for the dilute conformations of this kind of dipolar polymer chain. And then regarding the sequence, we found that if you have a more homogeneous distribution of the dipoles, or not just in dipoles but in general for any charged macromolecular system, if you have a more homogeneous sequence, it is more probable that the theta temperature will be lower for those kind of sequences. And with that, I would like to acknowledge my collaborators, Professor Muthukumar and Shafong, and also thank you for your attention, and I would be happy to answer your questions.
Hi. Thank you for providing that wonderful insight into synuclein. Just wondering, how would you try and correlate some of these results with its behavior in Lewy bodies in context of the disease?
So actually, in the context of disease, so these are not. So I won't say like these are exactly like amyloid fibrils. So I'm not sure like the whatever we observe in our system, we can directly like comment about the Lewy bodies from basically the condition that we are looking at. That we cannot correlate directly with the Lewy bodies.
But I would like to think that some of this affects the physiology of synuclein in the cell, right? Like its functions in vesicular trafficking, which is what it normally does.
Yeah.
So how does this interface with that? Just some general idea. I understand your focus is different.
Yeah.
But for me to understand as a biologist.
So what is known is so for particularly for alpha synuclein. So these simulations which we did was at neutral pH. But what is known for alpha synuclein is that if you go to a lower pH, that's when you see that it is much more prominent, much more easier for the amyloid fibrils to be formed. And at neutral pH is very difficult.
Hi. I was just thinking that for your RNA and protein interactions, you said that at lower salt concentration, I think you get interaction more with N-terminal.
Yes, yeah.
And then if you go to a higher salt concentration, then you in contact map probably you see more interaction with the C-terminal, right?
With the RNA.
RNA.
Yeah.
Right? That's what your observation is.
No, so this is the observation regarding the contact is that when you have RNA, you don't have contacts between the N-terminal and C-terminal, but more contacts between the N-terminals.
Okay, so it is always only with the N-terminal there is contact.
Yes. So what is happening is, if I go back to the, so because so since the complexation with RNA happens through the N-terminal, so all the N-terminals can be now closer compared to the C-terminal, and that's why you see a large number of contacts.
So this is precisely my point is, the point is that for this kind of RNA protein kind of interactions, people have found reentrant behavior. That means if you go to higher salt concentration, then might be the one which you are thinking repulsive, that can come into picture.
Yeah.
Are you seeing that?
So I agree with your point, but for the bulk solution, actually we didn't did a systematic study of the ionic strengths. So that's why. So this is I think so this is at 100 millimolar.
100.
So we didn't vary the ionic strength. But I agree with your comment that if I go to the very high salt concentration, I may also see this reentrant behavior. Yeah.
In your system?
So it will be, if I remember correctly, it's roughly around 6 millimolar or something like that.
Okay. And so the transition from the fluid to the percolated assembly, so this would be a second order phase transition or first order phase transition?
So this is more like a gelation in this case, actually, I would say. So this is a first order phase transition. So this particular case you are talking about, right? Yeah, no, so this is second order. Yeah.
In the case where you saw non-monotonic behavior, are you proposing that the RNA is causing nucleation and therefore the formation of intermediate sized clusters? Nucleation?
Yeah, so RNA kind of acts like the nucleation center, like basically.
So wherever there is RNA around it, the N-terminus of the synuclein chain
Yes,
comes and
but
clusters at
interestingly-
intermediate ionic strengths.
Yeah, but if you look, so for example, so for this one, the 20% one, you can see there are actually 2 peaks. So there are 2 peaks. So as I increase the RNA, it's not just the RNA which acts as the nucleation center, but actually also the protein chains also can aggregate.
So, but just to add, this feature that you add a third component and you see the nucleation concentration or CSAT going down is quite well known now. I mean, we see it with DNA and RNA, so it's something, and also in the physics literature, often people call it pre-wetting and so on. So it's so I think with other proteins. Yeah, thank you. So let's thank Shivananda again. And we'll go for lunch. So I don't know if you guys know. Oh yeah, we have to take a photo. Yes, we have to take a group photo. So let's go outside and